Hi students, this video it is in the continuation of the chapter reproductive system, and in the previous videos of ours we have seen the reproductive system in male and female, and today we will discuss for the spermatogenesis along with the structure of sperm. Spermatogenesis, it is the formation of a sperm which is the male gamete. It is the reproductive cell and haploid in condition, where the single set of chromosome it is present. Now, gametogenesis. Reproductive cells, they are called as gametes, and the formation of gametes, it is called as gametogenesis. And when it takes place in the male, the term is known as spermatogenesis, in which the spermatozoa is formed. So, in the process of spermatogenesis, spermatozoa is formed. Males start producing sperm when they reach puberty. Puberty is the state where the sexual maturity occurs with the help of the hormones, and sperm production it occurs in the testes of the male. The testes, they are called as the primary sex organs because the sperm, the male gametes, they are formed, and inside the testes the coiled tube-like structures are present, which are called as seminiferous tubules. So in the seminiferous tubules, the sperm formation takes place, or you can say the spermatogenesis occurs in the seminiferous tubules. For the detailed description, you can see the previous videos for the chapter. Now, Spermatogonia. These are the cells which are present in the seminiferous tubules. Single is called as spermatogonium and the plural is called as spermatogonia. Spermatogonia replicate by mitosis several times to form identical diploid cells linked by the cytoplasmic bridges, and these are called as primary spermatocytes. So here we used to say that some of the spermatogonium are the germ cells which are present inside the seminiferous tubules. Some of the spermatogonium, they differentiated into the primary spermatocytes. How the differentiation occurs? As they undergo mitosis. Now, the primary spermatocytes, they are formed from the spermatogonium, and after that, the primary spermatocyte, they undergo meiosis, right? So, in first point, you can say that the spermatogonium is differentiated into the primary spermatocyte, and after that primary spermatocytes they undergo the meiosis. And in this diagram you can see the spermatogonium after the differentiation, they are changed, they are converted into the primary spermatocyte. In the spermatogonium, the mitosis occurs, and what is formed? The primary spermatocyte is formed, and after that, the meiosis occurs. So, primary spermatocyte it actually undergo meiosis. As you all know that meiosis it occurs in two phases, meiosis 1 and meiosis 2. In meiosis 1, two cells are formed, and after that these two cells they undergo the second phase, and the resultant are four cells. So all in one meiotic division, four cells are formed. So here same, when the primary spermatocyte undergo meiosis first, two secondary spermatocytes, they are formed where the chromosome number it is reduced to half, right? So the primary spermatocyte, you have to consider here one point, that the primary spermatocyte, the condition is 2n, but in the secondary spermatocyte the reduction takes place and the chromosome number is half, haploid content, because it is the reductional division. We all know meiosis is the reductional division. So here the secondary spermatocytes, they are formed having the single set of chromosome, and now the meiosis second occurs, which produce the four haploid cells, and these cells they are called as spermatids. So this point you have to consider well, that in the meiotic first division, in first meiosis, the two cells are formed which are called as secondary spermatocytes, and they are having the haploid content, right? The primary spermatocyte, which is the first cell which undergo meiosis, it is having the diploid condition, but after meiosis first, the two secondary spermatocytes are formed having the haploid content. Now, after the meiosis second, the four spermatids are formed through one primary spermatocyte, and the cytoplasmic bridges break down and the spermatids, they are released in the lumen of the seminiferous tubules, and the process is called as spermeation. So this is the point of interest, children, that the release of spermatids into the lumen of the seminiferous tubules, it is called as spermeation, because sometimes the confusion occurs between the spermeation and the spermiogenesis. You know, after that, the spermatids, they undergo spermiogenesis. Spermiogenesis is what? It is the remodeling and differentiation into the mature spermatozoa as they travel along the seminiferous tubule until they reach the epididymis. So, spermiogenesis is the remodeling and the differentiation into mature spermatozoa. From the seminiferous tubule, they travel to the rete testis, which acts to concentrate the sperm by removing excess fluid before moving to the epididymis, where the sperm is stored and undergo final stages of maturation. So this carrying of the sperm from the seminiferous tubule, that you can see in the previous video of ours, because we have seen in that how the accessory ducts, they carry the sperms. But still, if you can recall, the sperms, they are formed in the seminiferous tubules, and after that, via rete testis, they come to the vasa efferentia, and then epididymis, and then vas deferens, which is also called as sperm duct. So this is the course of carrying of the sperm. So here this is written also that from the seminiferous tubules, they travel to the rete testis, which acts to concentrate the sperm by removing excess fluid before moving to the epididymis, where the sperm is stored and undergo the final stages of maturation. Because when we talk about the function of the epididymis, here we used to say that in the epididymis, the sperms, they are temporarily stored and undergo maturation, which is a coiled tube like structure. So if you want to have a definition for the spermiogenesis and spermiation, you can say that in the spermiogenesis, maturation of spermatids into sperm cells takes place, and spermiation, it is the release of a sperm cell from the Sertoli cells. Now come to the structure of the sperm, right? But before the structure of the sperm, I just want to have a quick look for the diagram, because now we are discussed for the secondary spermatocyte, we are discussed with the spermatid and the spermatozoa. See the different types of cell. This is what the spermatogonium in the seminiferous tubule, and after that it is differentiated into primary spermatocyte. How it is differentiated? By undergoing mitosis, right? So mitosis occurs in the spermatogonium. Primary spermatocyte is formed, which undergo the meiosis, right? And secondary spermatocyte is formed in the meiosis 1. So in the meiosis 1, secondary spermatocyte is formed having the haploid content, and in meiosis 2, the spermatids are formed. So these are the spermatids, right? These are the spermatids which are transformed into spermatozoa. So these cells, I just want to have a clear identification of the cells, because sometimes the labeling for this diagram it comes. So that is what. Now come to the structure of the sperm. Sperm, it is having the following parts: head, middle piece, and the tail. You can see the head here, right? Mid piece and the tail. Now, the head, which you can see here, this is the most important part of the cell, as it contains the nucleus. You can see the nucleus inside. Together, the two, acrosome and acrosomal cap, make the acrosomal region. So this is what the acrosomal region. It is formed during spermiogenesis. This acrosome, it is the product of Golgi complex, and it contains the enzymes. Apart from the enzymes, also contains polysaccharides like mannose, hexosamine, and galactose. So the enzymes which are present, they are very important for the fertilization. And this acrosome, it contains the enzyme along with the other substances like mannose, hexosamine, and the galactose. And the constituents of the acrosomal region, they help in the fertilization of the ovum. So the location is important, because the labeling of the sperm, it is equally important. So this acrosomal region, it you can see in the diagram it, and it is having the enzymes which are necessary for the fertilization of ovum. The middle piece which you can see, it is the central part of the sperm cell, and the mitochondria, they are present, and the mitochondria, they provide energy for the motility of the sperm, because sperm, it is a highly motile structure. So for that, energy is required, and the energy, it is being provided through the mitochondria present in the mid piece. Now this is the tail of the sperm, which is a thin elongated structure. It makes up 80% of the entire length of the sperm. Sometimes a centriole, it is also seen in the structure of the sperm, somewhat located here, and that centriole is there for the formation of the flagellum of the sperm. So this is what the structure of the sperm. and you can categorize the structure into head, middle piece and tail. So this much is for the spermatogenesis and the structure of the sperm. We would have different topics in the upcoming videos of ours where your queries are welcome. You can have the live interactive sessions with me. You can post your requirements as well. Keep watching the videos. Bye for now.